We'll start just does everybody know, start by introducing ourselves, and then we'll talk a little bit about the methods we use and the projects we're going to do. So I'm an evolutionary biologist. So that means I'm interested in studying how things evolve, where evolution is just a change in the heritable traits of a population over time. But I study evolution using mathematical and computational models. And the nice thing about these models is that they're transferable. So I can apply them to any system where there's an interesting question. So I've worked on speciation in spiders. I'm not sure if you can see out of curiosity, can you see my pointer on the screen? Or is that?
Yes, we got it.
And okay, perfect. So speciation in spiders. I've worked on plant pollinator interactions. I've worked on host parasite interactions. Urban pests. This guy might not look like an urban pest, but if you live in Chicago, he is. And I've worked on mate choice and the evolution of mate choice in fish, in birds, and in mammals, including humans. So why do we care about evolution? Well, one of the reasons is that evolution can help us to understand why things are the way they are. For example, this picture shows a male and a female zebrafinch. So the male is here on the right with the red beak. And an interesting thing about this bird is that juvenile females learn to prefer mates that look like their fathers. So this male has a red beak, so his daughters will grow up to prefer mates that have red beaks. But if we paint his beak yellow, then his daughters will grow up to look for mates that have yellow beaks. And this is kind of an interesting thing. Almost all birds do this. This is called sexual imprinting. Birds will do it for beak color, but for other things as well. And a lot of fish, some mammals, and even humans will do this for different traits. But learn mate preferences. If some traits are better than others, wouldn't it be better just to have a genetically encoded preference for the traits that work best? This is one of the questions that my lab has worked a lot on. And it's not today's topic, so I'm not going to tell you the answers. But it's one of the things we've looked at. Another reason for studying evolution is that it can help us predict how things will be in the future. So, for example, we know that climate change is altering the times that plants flower and the times that insects emerge from diapause. So will this disrupt plant pollinator mutualisms and lead to extinctions, or will these species be able to evolve so that the flowering times and the emergence states stay synchronized? Or similarly, humans are introducing species to new habitats all over the planet. And this is exposing new parasites or new hosts to new parasites. Should we expect these parasites to threaten the native species, or should we expect the species to evolve defenses to them? This is another one of the questions that my lab has worked on. So, Xavier, do you want to take from here with your introduction?
More precisely, I'm interested in the process of speciation, the origin of species, and the maintenance of variation. I also studied the evolution of non-random mating through the evolution of behavioral trait like habitat preference and assortative mating. Lately, I became interested in the evolution of life history traits, so how a species or an individual may change the way it grows, the time of maturation, in order to switch niche or remain in a niche that previously used. Just like on the right-hand side of this picture here, we have two alewives that are the exact same species, except that at the bottom we have individuals who choose to stop their growth and remain at the size of a juvenile, but while being mature, and they're feeding on smaller organisms that the individual that continues to grow at the top and basically changes niche as it age and only mature later. And those have interesting feedback phenomenon between the resources they feed on and the choice they make when they decide to mature at small size or at larger size. For historical reason, my study system are fishes, but in reality, I only do theory, and I'm behind a computer most of the time. But we're not here really to talk about our study system and the system we studied in the past. We're here mostly to talk about the tools that we are using in order to solve our problem. And the tools that we've used in the past to solve our problem can also be useful in this CURE project. One of those tools is ordinary differential equation that we will write the shorthand from as ODE. So what are those things? So if we think we have one OTU, so those how do you call them again? Organismal unit, or a bacteria population, the x will be its population size. So we have d, which represent the change of the population size, that we divide by dt, which is the change through time, and it's function of something. So we study the change of the abundance of a species through time. So a species can be simplified to the extreme by having a birth rate that we will represent by B. So the more individual there is that reproduce, the larger the increase. So the dx over dt, the rate of change, will also increase. Those same individual can also die, which is represented by the term M here, that is their mortality. So a fraction of the individual will get out of the population, and that will decrease the overall population size. If we do a little bit of arithmetic and put B minus M as a factor, and then replace it by R, that we call it the growth rate, we have a really simple ordinary differential equation where we say that the change of the population size through time is function of its growth rate. And this growth rate is simply the birth rate minus the mortality rate. So if we make a forward modeling of this, if we plot it on a graph, we have a density that will increase through time, and it will increase exponentially, meaning that as time goes on, we'll have more and more individual of that species, and that will continue forever. But in reality, if the world was like this, the first species to come to Earth will grow at an enormous rate and end up covering the entire surface of the Earth. So obviously, this is not the only factor that is occurring here. So what can constrain population growth? The first answer to that question can be intraspecific competition. Here we have the same exact ordinary differential equation as before. Our dx over dt is equal to r multiplied by x. But in this case, we add a limiting factor here to have a logistic growth of 1 minus alpha x. Alpha represents the competition factor between two individuals. So basically, we have a pie of fixed size, and the more individual we have feeding on that pie, the smaller the piece for each individual will be, and therefore, the smaller the amount of resources they can acquire, the smaller their growth will be. So if we plot the result of that equation, we have at really low density, so when x is really small, we get basically the same equation as before, r multiplied by 1 multiplied by x, which will be an exponential growth rate. But as x get closer and closer to 1 over alpha, we get closer and closer to the equilibrium point. Because here, if x is equal to 1 over alpha, the term in the middle will become 1 minus 1. And this will be 0. So multiplying whatever with 0 will be 0. So our rate of change will be 0. So the population will be at a stable equilibrium. And we can see that around that population tend to reach a certain equilibrium and stay there. And it can be because of intraspecific competition. So here we have competition among conspecific only represented in alpha, but the world is composed of more than one species, and those different species can also compete among each other. So here we have the same equation as before, and we add a little more at this step. So we still have dx1 over dt, so that's our first OTU, and its growth rate is r1 multiplied by x1. And the limiting factor that we have in parentheses is once again 1 minus alpha x. That was our intraspecific competition before. In this case, we put alpha 1, 1, say that it's in extra-specific competition factors. So species 1, the effect on species 1, and the other term is the effect of species 2 on the growth rate of species 1. So here we try to have a correction factor. For example, if species 2 is twice as fast at eating its pie or acquiring the pie, one individual of species 2 will be equivalent to 2 individual of species 1. So the alpha 2 will be twice as large as alpha 1, 1. Or if it's the opposite, it's not as good as a competitor as species 1, then 2 individuals will be needed to have the same impact than 1 individual of the same species. And if we plot this, we realize that in most cases, we don't have coexistence. In this case, one species is a better competitor than the other, and it will drive the population of the other species down up to extinction. But if we go into a multidimensional resource model, that it's not really hard to do, but it's for illustrative purposes, we stick to one resource here. A lot more possible outcome of coexistence are possible. Another thing that can constrain population growth is predator-prey dynamic. In this case, we have, again, an OTU of the prey will be species 1. So we have the growth rate of the prey, dx1 over dt. That is function of his intrinsic growth rate that we called alpha in this case. And what limits his growth, the minus factor here, x1 multiplied by x2, is the amount of encounter that the prey has with the predator. And beta is the attack rate of the predator. So the more the predator attack, the more limited will be the growth of the prey. And if we go down to the predator growth rate, the growth rate of the predator, it doesn't have an intrinsic growth rate, it's function of the encounter it has with the prey, so x1 multiplied by x2, and the delta is the conversion factor that says how many prey do you need to eat in order to make, let's say, one new predator. And the predator will have a mortality gamma here that is multiplied by x2. So those two ordinary differential equations are coupled together because x1 is function of x2 and x2 is function of x1 over here. So the usual dynamic we have for coexisting predator-prey is an oscillatory behavior where we have, when the predator is rare, an increase in prey because they are not limited in their growth by the predator, so they have an intrinsic growth rate that help them grow and reach higher number. But then with a large abundance of prey, the predator can feed really easily on them, and there's an abundance of resource, so it will increase in size. But when the predator is at its maximum, it's eating a lot of prey. So the prey is going down fairly rapidly. And when the prey are rare or become rare, the predator will struggle as well to find those prey. So his population will go down. And this cycle will repeat over and over where we have two oscillations that are not in phase. So the peaks are not at the exact same time, but the predator peak is slightly after the peak of the prey. So we have abundance in prey, later on we have abundance in predator, and then we have abundance in prey again, and it's cycle over and over, just like we have in this phase plane representation of the same thing. On the y-axis we have the rabbit population size, and on x we have the fox population size, and we can see that it always turning on the same trajectory, so it's a stable system. And the importance of it here is for cure, the prey or the predator can be phage and viruses, and our prey can be the bacteria of our microbiome. So in order to simulate this entire thing, or the microbiome of the respiratory tract, we need to have several species and several OTUs for growth and their competition and their predation. So we know that the equation we have are scalable to more than one species, and it's fairly easy to extrapolate. So Now we know there's intraspecific, interspecific competition, and we have predation. But how do we estimate the importance and the effect of each of those factors? And now I'll switch the screen to Tucker, please.
So the way we're going to do, so Xavier introduced the kinds of models that we're going to use, but then the importance of the parameters in the model for telling us how these systems behave. But now we want to talk about how we're going to get the parameters, how we're going to estimate the values of these parameters. So let's look again at the system of ODEs that Xavier described for competition before. So we can see that this system has several parameters in it. It's got these R's, which are the growth rates that Xavier talked about, and it's got these alphas, which are the competition coefficients. And depending on what these look like, depending on what these values are, that will tell us what this trajectory, this set of trajectories will look like. But how do we get these values? Well, we can represent the values as these in series in vectors and matrices. So this vector here would tell us, this vector of R's will tell us the growth rates of all the OTUs in the system, and this matrix of alphas will tell us how each OTU competes with every other OTU in the system. Right. So the way that we get these is the same way that we'd find parameter values for any other kind of model. That is, we measure the response variable, we measure the predictors, and then we fit a model to the observations to estimate the parameter values. So in this case, the response variable is this value here on the left. So it's the rate of change, this dx dt. Right? So it's the slope of the population trajectory at some point in time. The predictors are the densities of each population at that point in time. So that is, they're the values of x1 and x2. So if we have the value of x1 and x2, and we have the value of dx dt, then we can put these into the equation and we can solve for the values of r and alpha that make this equation true. That's how we fit a model in general. But why should we bother with this? Well, after all, because we know how the system behaves, right? We had to observe it to get the parameter values. Well, the answer is that if we have parameter values, we can make informed predictions about how the system will behave when it's in states that we have not previously observed. And sometimes these behaviors are unintuitive. For example, in the system that I'm showing you here, right, this orange population outcompetes the blue one. Right? So we have both populations at the beginning. The orange one persists, and the blue one disappears. But if this system is disturbed, for instance, maybe there's an illness that allows the blue population to spike, right? Then the whole system can be upset. If the blue population spikes like this, the orange it will outcompete the orange population, and the orange population will never recover. The system will never go back to the way it was before. And if the old state was healthy and the new one is unhealthy, this is a real problem. So this is up to this point, these are really the sort of the basics of the model. Do people have questions at this point? Because we'll go on to talk about some of the specific challenges that we think are related to CURE. But questions about the modeling approach at all? Yeah. So, and if you feel like we're mildly obsessed with this, you're probably right. And this is why. Right? For every data point in our data set, we need to measure both the predictors and the response variable. And the predictors in our measurements are just the densities of the OTUs in the system. So that's the how frequent each one is in a given sample, and we'll get those directly from the sample. So it's I want to say it's easy. It's easy for me because it just comes in as one data point, and I know this is a hard part for sequencing, but from our point of view, data-wise, it's pretty easy. However, the response variable is a change in density over time for each OTU. And to get a change in density, we need two samples. Right? We need the initial density, and we need a final density. So 2 samples make 1 data point, or 3 samples could make 2 data points, or 4 samples could make 3. But we need this gap for each data point that we're going to get. And the time between the samples has to be right. And the reason for this is twofold. First, when we're measuring the density of each OTU in the system, we're not actually getting the exact density. We're getting the density. And if the sampling period is too short, then there won't be very much time for there to be a real change in the density of the OTU that we're interested in. So what we'll pick up is mostly sampling error. So, for example, in this case, with these two heuristic points that I put here, we would infer that there's a downward trajectory in this OTU when actually the trajectory is upward, simply because the sampling is so close together that error has swamped the signal. Alternatively, if the sampling intervals are too far apart, then the rate of change over the sampling period won't be constant, right? And then the slope that we measure will be incorrect, right? So in the extreme case, so like here, the slope that we would measure if the second sampling time were too far is actually too far away is actually much more shallow than the true slope, right? And in an extreme case, the measured slope would be completely unrelated to the densities at the beginning interval, and again, all we would get is noise. So we have to sample at the right interval. But what is that interval? And unfortunately, we don't know. And we won't know until we have some data to look at. And this is a challenge. Now, there are a couple of solutions to this. And the first one is the sampling system that Nikos has already proposed. Right? So for a subset of our study population, we would take a few short interval samples early in the study, and then longer interval samples as the study goes on. And hopefully, at least some of these would be at the right interval, would be at the interval we can use. So that would be one way that could help us with this problem. A second possible solution is more ambitious. So in this one, we would use simulation models to try to predict the optimal sampling interval. So for this, we would simulate a dynamical system with a diffusion process that just adds error to the system. Then we simulate sampling from that process, and we fit a model to the simulated samples. And then we ask, can we use the simulated samples to calculate the optimal sampling interval for the system? Because if we can do this, right, if we can predict the optimal sampling interval for a simulated system, then we can use the same method to predict the best sampling interval for a real system. So I have a very good student who's working on this problem right now, and hopefully within the next few months, we'll have a method set out that will help us do this. And if this works, we'll be able to use our samples from early in CURE to then predict optimal sampling intervals for later. So will these completely work? I'm not sure. If they do work, will it be possible to get the samples in the interval that we want? We're not completely sure of that either. If we want samples every 30 seconds, it's just not plausible. But this is the best we can do until we have more information to work with. And I'm optimistic that we are going to be able to get samples in the interval that we want. People who've done this for gut microbiomes have estimated that the best sampling interval is probably around 2 days. They don't know either. They're guessing. But I think at least some of our samples will have at that period. So I think this is plausible. The second challenge, the second challenge that we'll face here, and this is one that that came up a little bit in Nikos's question, is figuring out how to get enough data to do what we want to do. So if we think back to the very simple model that we used to introduce the ODEs, we had just 2 OTUs in it. Therefore, we had an interaction matrix with 4 parameters. Right? We had the effect of OTU1 on OTU2, the effect of OTU1 on OTU1, and then the effect of OTU2 on OTU1 and OTU2. But more generally, we'll have one interaction term for every OTU on each other OTU in the system. So like I said when I was answering Nikos's question, if there's 380 OTUs in the upper respiratory tract, our model would have about 144,000 interaction terms. And a rule of thumb is we'd like to have about 10 data points for every interaction term that we need to estimate. So we'd like to have about 150 million data points. And obviously we're not going to have that. So I should start by saying this sounds worse than it is. The first helpful thing to recognize is that each of the pairs of samples that we get isn't really just one data point. Instead, it's one data point for every OTU that appears in the sample. So if a sample has 80 OTUs in it, then a pair of samples is going to be 80 data points. So it immediately makes the problem order of magnitude easier. So that helps. In addition, we can make this problem easier using the method that, or using the idea that Akis brought up a few minutes ago. Right? That is, we have some a priori knowledge of what some of the OTUs in the system do. Okay? For example, some of the OTUs are phages, and we know that phages don't attack other phages; they attack bacteria, and specifically, they attack. And for many of them, they attack particular families of bacteria, and we may know what those are. Right? So for the things that the phages don't interact with, we can begin to populate this matrix of interaction coefficients with zeros. We might be able to do the same thing with some of the bacteria if we know that the bacteria are using different substrates. If we know that some kinds of bacteria don't compete with others, we may be able to put a set of zeros into these matrices that will make fitting this matrix a lot easier. And we may also be able to leverage some information about the phylogenetics of these bacteria. If we can assume that, if we can make an initial assumption that bacteria that are more closely related have stronger competition coefficients, we can begin to populate this matrix on with an assumption that says that the strength of competition is related to the phylogenetic distance between strains, and then instead of needing to assign a single parameter to every combination of to every OTU by OTU interaction, we can assign a small number of parameters to describe interactions how interactions depend on phylogeny, and that may greatly reduce the number of parameters that we need to estimate. Finally, the last benefit, or the last important thing to recognize here, is that we don't have to estimate all of the non-zero parameters in the system. We only have to estimate the ones that matter. And there's some well-established methods for picking out which ones those are. So we can use information theory approaches, we can use lasso approaches, and these are both things that my lab has done quite a bit of. Or we can use machine learning and cross-validation, which both Xavier and I have done in the past. In either case, the basic idea is the same. We identify and estimate the parameters that have the biggest effect on the system, and we ignore the ones that are too small to matter. And then we ask if the parameters that we've estimated allow us to predict the way the system behaves. And if they do, we've gotten what we need. And I'm fairly optimistic that we can do this based on the results of the rat gut study that I mentioned earlier. Ridenour and colleagues were able to predict the behavior of the rat gut microbiome fairly well by just estimating about 6,000 parameters. And that seems like a plausible number for us to estimate. They did that with just 36 data points, 36 sampling points, and they got about 6,000 parameters. So they did a pretty good job of estimating parameter values, and we might have a few tricks that we can apply that they didn't use as far as a priori knowledge of the system and phylogenetics. Okay. So I think shall I let Xavier take the next section, and then we'll pause for some questions again towards the end. Okay. So
Dora, can you switch the presenter screen, please? Thank you. Okay, so what we will measure over here is a combination of the beautiful internal dynamics that our mathematical model are picking up, but also some external factor like the presence of cat, the sampling error, and the noise. And just to illustrate this problem that I don't have a solution for, is just a fact of life, we can take the Lotka-Volterra dynamic that is a fairly stable oscillatory behavior that is repeating through time, and adding a small amount of noise can change qualitatively quite a lot the behavior of the system. It goes from pure oscillatory behavior to a lot more than just a perturbation of that clear strategy. but still both species can survive, and to some extent, in this case, it looks a lot more stable. We know we have an average of rabbit of 25 and an average of predator of 10, and it moves around that. But that's something we will have to keep in mind for our data collection, is that we're having a lot more than just the pure internal dynamic. And another thing that will be really important is the potential variability among individuals. So different individuals may have different microbiomes, so they may be different environments, so their environment can have an impact on all the alphas that we previously estimated. So those alpha may be individual dependent, or at a larger scale, maybe there's some geographic effect, the presence of bacteria in some part of the world that are not present in others, or there can be a sex effect. Some hormones that are different in different individuals may actually affect the competition or the predation within that microbiome. The age, a middle-aged person with a strong immune system may have a different microbiome dynamic than a newborn or an elderly. So we go from that potential 144,000 alphas that we would need, and we need to multiply that by all the different groups we can partition those differences. So the number can become extremely large, and it will make our task really difficult, because if we try to estimate a single alpha between two groups that have clearly different microbiome because of reason that we know or we forgot to take into account, then it's gonna make our estimate of alpha a lot weaker. So if you know about any a priori information about the microbiome of different individuals that we can put in our model, so we could have, instead of having a single matrix of alpha, maybe two, because the effect of sex is really important, or we could have 3 or 4 age category, and that will make our estimate of alpha, all our parameters, a lot easier, because we won't have that other confounding factor. Obviously, we cannot have individualized medicine here, where every single individual will have enough data point to get his or her own competition matrix. But if we can get an effective method to partition it the most effective way, it would be really good. So just keep that in mind for later on. We would really like to know if there's potential a priori clustering that we can do among the individuals. So now we're going to switch gear a little bit. We've been looking at how our model can fit the data by adjusting the parameters. So let's assume we're a few months down the road now, and we have our model and we have our parameters that can fit those data pretty well. What we can do with those models and parameters is to predict what's going to happen in the future. So take a more forward-looking approach, some forward modeling. So if we have a state of a system of an individual that is in a crisis, in an asthma crisis, and we know what is the target of an healthy state, we can take our model and our parameter and do an in silico experiment, trying to find the shortest path from that unhealthy state to the healthy state. And we can all do that in a computer by using our model and our parameters, and then we can potentially have a treatment for that. And there's different way to do the forward modeling. One way is the stochastic method, which is more or less a random walk on the health fitness landscape, where we test different points, and when we find a better one, we climb to that point, and then we start searching around that point again, and we basically climb that that health fitness landscape and potentially extract a treatment from it. There's also deterministic method where we can look at a gradient in that same hypothetical health fitness landscape and find the gradient that will give us the most effective response. Just like climbing a mountain, you look around and what is the highest point around me, then I'll take a step towards that point, and you start over and over. And if the fitness landscape is not too rugged, then you're going to towards the optimal. But that's really in a few months when we will have our model fit with the parameter, the good parameter, and then we will be able to do some forward modeling and hopefully try to find a path where we can try to adjust the amount of bacteria or phage, or try to reduce them using antibiotics in order to improve the health of the patient. So far we presented a few things, but we still have a few questions that are burning in our brain. One of that is point number 2 here that we briefly mentioned. So any data that you would have that could help us partition the data a priori, so known information about non-competing OTUs or seasonal differences or regional differences that could help us not trying to estimate a single alpha when there's in fact 2 because of those other factors. Another point that we kept out of this entire presentation is the external factor, everything that is outside the sampling point that we do in the nose of the patient. So the patient still have his lungs, and thing can move from his lung to its nose when he's coughing. So maybe there's some input of new individuals coming from there, and maybe the input is large enough that it can actually change the dynamic. Or everything that comes from the environment through the nose can also have a potential really big impact. So we would like to be recomforted that the main effect here that we will be looking at, or when we will search, will be the internal dynamics and not an external forces where we have absolutely no sampling power, for example, deep down in the lung or outside the room where the person sits, for example. So I would like to open the floor for discussion at this point to get basically any idea about that.
I can start before Akis. I can see Akis that is ready to give some answers, and yeah, we have first of all we have considered these questions, and in relation with the first one, it seems, without being the final answer, but it seems that the nose basically works like a sieve. So we do get, when you do get lots of input from the outside, but however, most of the times these are sort of small numbers, and then the microbes move from the upper airway towards the lower airway, and they are not changing much, at least in health, they're not changing much there. So basically you have less and less microbes from the upper to the lower airway, and if what you said happens, so when you get something back, it's usually of the same kind as it was coming in. So you won't expect to have a major influence from the lower airway. Of course, this may change to some extent when you have a specific pathology. So if you have a pneumonia, if you have something where you have microorganisms that develop in the lung, then you might change, so them coming out, you can change the balance, but it will be quantitative, it will not be qualitative, it will be the same ones that have multiplied. So, I mean, we have, let's say, a rather good system. It could have been much more difficult, but we do need to take into account that we have all the time microorganisms coming in and out. So I don't know if Aki, you want to add or give more details about that.
Yeah, so in terms of being a little bit more practical, we know from, at least for bacteria, because for phages we don't really know anything regarding your question, but things we've learned from the Human Microbiome Project or from the few publications that are out regarding the respiratory tract and the nose, we know definitely that if we create a gradient regarding the absolute number of OTUs per species, we can definitely, let's say, discard this noise that might be coming in from the environment. And this is mostly based in the observation that the species that are, let's say, most commonly found between people or between published studies are the ones that are always have the higher abundance and the higher absolute number of density in the microbiome. So I guess by picking, let's say, throwing out or creating a threshold in the level of the number of reads we're getting per species or per OTU, we can perhaps discard this variability that will come from the environment.
Okay, that's good.
Now, in relation to the second question, I think that this is also, I mean, I will answer to you, but I'll also make a question. We have, so among our plans, and this is what's already happening, and Akis is working on this at the moment, we are going to have information about geographical variation. And of course, we can have also information about seasonal differences, gender differences. So we have data already on this. Now, my question is, when we do get some type of differences between groups, would we need to make separate arrays or separate matrices for each of these parameters, or is there a way of trying to correct or in a way introduce this factor into the model? Because as you mentioned, there are several parameters. We know that each parameter will have some kind of effect. So what is the way of dealing with that?
We could use one approach would be to take a system that for one group that we're able to parameterize fairly well and use that as our null hypothesis for parameterizing the second group, and then we may be able to identify which parameters differ, and that would save us from needing to parameterize completely separate models for every group. Parameterizing completely separate interaction matrices for each group of people would be a worst case scenario, and hopefully we wouldn't have to do that. It seems more likely that we would have to that we might have to change the maybe the growth rate functions, or the growth rate, not the growth rate functions, but the growth rate parameters, which is quite a bit more doable than doing the whole interaction matrix, or identify specific points in the interaction matrix which might differ, and those are again probably doable with the amount of data that we will have, as long as we have enough data from each group. If we have just one person from a group, we won't learn anything at all about them. But if we have a roughly even split of men and women, I think we have some potential for parsing out the differences that are attributable to sex. Does that help? Is that the...
Yeah, so this is the direction of my reasoning. But the other thing is that when we do have the data at the moment to be able to compare between gender, for example, between sex, and also in terms of season, as examples. But then when there are differences between genders, for example, or between seasons, then one needs to, at least in the statistical approach, you need to see how much of any of these risk factors would influence the other, because it might be that more males have been sampled in spring. So these type of other third level interactions do we need to actually, hypothesize that there are X different categories and then include them in such a model, and then use this information to parameterize your models? Or again, there is no way, or then we have to do it somehow separately? Because otherwise, in the end, we will end up having each individual being different from the other one.
Right. It would be really helpful to know which categories we think are going to be most important, because like you've just said here, that if we use enough categories, every individual will fall into their own, and then it will look like category describes everything, because the category would essentially everyone would have their own category. But if we know what categories are important to begin with, if we can guess that sex is more important than age or city of origin, then we can focus on that and make some and test whether it has an effect. But the more a priori information we have about what's important, the better it will be. We can account for the fact that if we know that two factors are important, we can account for unbalanced sampling. So the sampling more men in spring versus winter. Those are things we can deal with. The unbalanced sampling shouldn't be a problem, as long as we know what things we care about.
Okay. Another challenge. Sorry, Nina, please.
Yes, I wanted to ask you, shall we take into consideration the effect of macrophages, epithelial cells, and so forth? Because the phages, our phages, bacteriophages, they may absorb on the surface of macrophages as well, and some part of population may be lost. Should we take into consideration these facts as well?
Okay, I'm not sure I completely understood the question.
The question is that when we are modeling, where we're talking about the prey and predators, and also the background, such as noise. For example, you said that cat dust and so forth can influence the model. So I'm talking about the immune system, the immune cells, for example, macrophages, T cells, and so forth, which may be attacked by the phages erroneously, by mistake. And in this case, we will get less bacteriophages around than we expect to have.
Right. Okay. Okay.
Is that common, or it's uncommon effect?
Actually, according to the literature, this happens. Usually this happens, and if bacteriophages are adsorbed on the surface of the T cells, they strike the immune system, the general response of the immune system, which is another factor which I think could be taken into account as well. I don't know. So this is quite a complex, really very complex microbiome and environment in which the phages will act.
So those phages are the predator, and they have in our model an intrinsic mortality, and that phage attacking the immune system may be part of that mortality, because my understanding is the outcome, they won't be able to reproduce through attacking the immune system. Is that right?
Yep.
Yes, that's right. Yes, that's
okay, so
right.
we can include that in their intrinsic mortality then.
Okay.
Yeah. And then as they cause the immune system to spike, then there's the immune system. The immune system will attack other things, though, since it's spiking. Is that correct? Okay. So
I'm having a...
right. Okay.
That we will have some information about the immune cells and perhaps immune status through secreted factors in these patients but only at specific time points. So this is something that we cannot sample very often. Right. Within this first phase, we're actually going to have just one sample from this, but then perhaps if we see important differences in relation to the immune effect, then we might consider repeating the sampling or changing the model in relation to that. But we will have some information. I'm not sure how we can integrate it, but we will have, let's say, the number of different types of cells and the concentration of different cytokines. So this is a type of information. We can also have some transcriptomic data we had, but not very early, and then see whether these parameters are in any way associated. But I'm not sure how or whether we will be able to include this data in the model. At some stage in the future, we said that we're going to try to identify interactions, but not necessarily for including them in the model, but for identifying the different interactions, describing the interactions between the immune system and the microbial communities.
Yeah,
Can I add? Yep.
Go ahead. Go ahead.
Okay, just if I can add something regarding the a priori partitioning of the data. So, okay, definitely age will be a factor that we expect to play a role. Even though everyone is talking about seasonality, there is only one study in the upper respiratory that has shown a difference between seasons in bacterial OTUs, and that's in that study it wasn't based in longitudinal data, so they didn't check the same sample during the year. Apart from that, if we take into account that at least, I don't know, a bacterium that is specifically found in spring or in winter, I don't know if I'm mistaken. So even though regarding eukaryotic viruses, we know that they follow a certain seasonality, regarding bacteria, we don't know that something like that happens. So I wouldn't put seasonality really high in this list. Instead of seasonality, I think the treatment protocol that the asthmatics follow can have a detrimental effect both in the microbiome, as we've seen in our own data, not only in bacteria, but also in the phages. But this would be a variability that will be incorporated in the system. So we should categorize or stratify the patient, I think, based on their treatment. This should be really high in the list.
Okay. Yeah, that makes a lot of sense. So I think, yeah, especially since one ultimately will probably want to make predictions for interventions in the background context of treatment. So yeah, I think treatments. Yeah, I think that's a good idea. Also, briefly back on sort of Nina and Nikos's question, it sounds to me like Xavier's answer about capturing the being able to capture this effect in the intrinsic mortality rate of the viruses is probably true, and the boosting in the immune system probably will then also be captured in a boost of what looks like an attack rate of the virus. So our model will, in our model, it will look like we won't be able to see that it's actually the immune system that's killing whatever it's killing. It will look like the viruses are doing it, but the model should still be able to capture the effects. So I don't think it will weaken our predictive power. I think it will just give us an unclear picture of the exact causality of what's going on. Yeah. Yeah. Treatment. Yeah, I think treatments are. Yeah, I think treatments are going to be a super important thing. Okay.
Yeah. I do have another couple of points which are relevant to some extent, because we're talking about OTUs and abundance as the factor that we are explaining, we are evaluating. But do we actually, so first of all, you can do that at different levels, whether it's species or it's genuses. So actually it can make a difference on what level we evaluate that. Some of the methodology that we use in the sequencing, again, can have an impact. And also there are more complex parameters, like, for example, relative abundance or diversity or things like that. Do we know that there is a specific parameter that is the optimal for the modeling, or because then, and that's in relation to what we're saying before, then we can have this parameter in a simple logistic regression model with all the possible effects of age, seasonality, treatment, gender, whatever it is, and then find out which of these parameters are really important, but in relation to that specific one, and then go back and use these parameters to informing our further work. So what about these different characteristics of the microbiota? Is this just the OTU, and at which level do we assess that?
Yeah, so that's largely an empirical question. So we'll want the fewer OTU.
I'm sorry, I lost you. Tucker?
Okay, maybe I can try to chip in here. I cannot hear Tucker either, but basically we need to have a level to work with, and my understanding was that we would get data from OTU and we can do our analysis on the data we have, and then there was no indication that we will get species specific or other information to analyze the data. But if we do have another layer of information, we could take it into account, but then it would increase the number of parameters because we would have interaction among species instead of OTUs.
Well, species and OTUs are not very different. So OTUs is operational taxonomic units, and then you can use it for different levels. So because exactly it is not exactly the same for each microorganism, so it's actually quite complex at this level. And we do decide which one we are going to use, or we're using both of them. So, and of course, the more specific you become, the more you have, let's say, more different species or the different groups. But then it's difficult to make groupings. And the higher you go, you have a more sort of general overview and less family. So I don't know, Akis, if you want to comment on that. And
yeah, so it's a difficult question to answer. So first of all, whether we're going to get sufficient information at the species level depends a lot on the resolution, and the resolution depends on the depth of the sequencing and how enriched the initial sample is. We can definitely use different taxon levels to analyze the data and compare do the comparison between groups. The most usual is people stop at the family level, or they move up to the genus level the most. Where the more deep you go in the taxon classification level, the more information you lose. And you become more specific, of course, regarding certain species. I think this is something we can agree or discuss about it in another meeting, perhaps, because there are many parameters that we need to take into account. But it's definitely a very good question. I mean, we need to check this.
And this is in particular in relation to the phages because of their specificity.
Yeah, for the phages, for example, that's
about a phage family. But then when we're talking about interactions, and we know that the interactions of the phages are rather specific, then we need to decide whether what we find at the family level can be relevant, or how relevant it can be.
Yeah,
we might need to make a priori decisions on that. So we just I just did a model recently for a very different problem, or for a very different question that addresses sort of the same issue. One technique we might be able to use here would be to start with a full set of OTUs and a phylogeny of how they're related, and then do a clustering that's actually informed by the data. So essentially merge the closely related OTUs and ask if, so by doing that we lose a lot of parameters, we lose a little bit of predictive power, and then use information theory to fall back until we have the set of of OTUs that best describe the model. And some of those might turn out to be at the species level, and others might turn out to be at the family level. And we could let the data itself answer what's the best way to do that.
Yeah, that would be good.
Mm-hmm.
Yeah, I think David should be in this meeting as well. He could provide some answers. In general, when we do metagenomics, it's not usual to use OTUs because exactly it's the aim of shotgun sequencing, not to use OTUs. So yeah, we have to see what we use.
So how do you break up the groups then? If it's not OTUs, it's by genotypes then.
Yeah, we're having the genotype information, and we have the taxon classification information.
Okay.
Because operational taxonomic units are usually an arbitrary system.
Okay.
Yep.
Most studies that use OTUs are based in 16S based sequencing, which of course the samples are pre-amplified.
Okay. Yeah, we should chat about this because I think there might be something neat we can do here with finding the optimal set of well, whatever we want to call our taxonomic units, based on the demands of actually describing the system. I've never seen it done, but I think it might be really fun to try.
Plus, I think if we start from, let's say, an analysis at the genus level, and we observe, let's say, 2 or 3 facts or interactions between different species, we can use the higher order classification in order to verify our results, and also check whether this is a general, what we are observing, something general that it affects, let's say, a certain bacterial family or a certain family of phages, or rather it is very specific and goes just to the species level, and this is some really specific for, let's say, 2 things.
Mm-hmm.
I have another challenging one, which, I mean, challenging for me. So, do we know that actually there is an optimal sampling period for the whole community? Or that might be that there can be an optimal period for a specific microorganism or family, and then, you know, the rate of change of another one can be different, and so the optimal time for another one, it can be different. So are we referring for the optimal, let's say, sampling frame for the community, or this is based on, you know, the assumption that the dynamic will remain the same within different sampling frames?
So the reproduction time of every different bacteria and phage will be different, therefore the internal dynamics will be different. So there's no universal optimal solution. The thing is, we need to capture the main dynamics, so the main driver for those asthma crisis and everything. So we should focus on those, but my understanding, we don't know exactly which one are the most important yet. So we're a bit shooting in the dark now by trying to find an optimal. But I think last time we talked in Greece, we were not even sure yet about if there was those cycling dynamics or what would be the period of them. So we need to get that information before actually being able to answer that question about what is the optimal. And it's going to be a specific thing for this study system.
So the way that we have designed our initial sampling frame for the cohort is so that we have a number, at least 3, I think, but a number of samples within different possible rhythms. So, or a circadian one or a weekly one, or, you know, so that we might try to see whether there are such circles or not. Is this something that you need to do first? So is this data that you can use to try to identify whether there is a circadian or a weekly or whatever rhythm, and then go back and decide whether these, the next sampling should be done on the same time of the day or the same day of the week, etc. So that's what our hope is.
Mm-hmm. Yeah, I think so. The sampling plan that I've seen where you're looking for both, where we're looking for both the circadian and weekly cycles, I think will be quite useful, because if the cycles are there, we'll at least have a we may not be able to fully characterize them, but we should be able to recognize that they're there, and that would then tell us whether we should be whether we should try to sample at the same time of day, same time of week, or not. So that I think will be quite helpful.
Yeah. That's great.
And in the best case, so we're focusing maybe on a harder case scenario. Maybe we have only two states of the system. One is healthy and the other one is not healthy, and they are stable to a great extent with a quick transition between the two. And then having a close sampling will tell us also that this is a stable state. And we don't need all those sampling, but intuitively it would be surprising that we have that many species coexisting and preying on each other that would not result in some cycling behavior or temporal variation.
I know, but so we're coming a little bit back to what we were discussing before about the specific species or what. So it might not be that a species is important, it might be that a family is important, or the metabolic activity of these microorganisms might be important. So you might see a lot of diversity at one level, and then you have much more uniformity and much more meaningful interaction at another level. So I think we will need to try to dig into that, because it might be so we know that the sets of microorganisms in each individual are quite different, but there must be something which is stable within that. So I think we will have sort of iterated rounds on what we are going to be the input to the model, and hopefully we'll be able to do that without having to change the model too much.
Mm-hmm. Okay.
Okay, are there, do you continue, or are we all ready to do questions from the audience?
Yeah. We're at the question for the audience, I think.
Yeah. So we'll definitely. I know we've kind of been taking questions. As.
Okay, so are there any other questions or thoughts or things what people want to share? I'm sure that you all understood everything and that you all know what a lasso approach is, because I don't. So if there's another question, Tucker, what's a lasso?
It's least angle squares regression. Basically it's kind of a cool way to fit a model where we don't know. The normal way that people fit models is to try to estimate all the parameters and minimize and use those to fit. If you think about it, fitting a line through the data, you're minimizing the distance to the distance from your predictions to the real data. In a lasso approach, instead, what you're doing is you start with assuming that all the parameters have value 0, and you look for the parameter that is most strongly correlated with the response variable, and you give it a small amount of value, and you keep and then you check to see then that will change the residuals in the data. So then you look for the residuals that are still most correlated with whatever predictors are left, and add a small amount of value to those, and we keep adding value to predictors until we reach some level that gives us the maximum predictive power. Because at some point, when the model is over parameterized, you start to predict things that aren't true anymore. So basically, it's a fitting method that minimizes the total parameter value as opposed to minimizing sum of squared error. It was worked out by Rob Tibshirani and people in his group about 15 years ago now, and it's really taking off in a lot of fields. So it's a pretty powerful way of picking out parameter values. That's will be fun to apply to a system like this.
Okay. Okay.
Can I add something?
Mike.
Okay, so there was a recent paper that showed that in regarding phage bacteria interactions during an infection. So over there, they demonstrated that at the population level, the immunity of the bacteria in the beginning restrains the replication of phages until the bacteria reach a very high number, and then they lose, let's say, the phages in order to increase the chances of the phage to go into a lysogenic cycle. So instead of killing the cell, actually being incorporated by it. And I was wondering whether this is something that can affect any of these models of prey-predator interactions.
So the phage is incorporated. So it's more dangerous than when the bacteria is common than when it's rare. Is that what you're saying?
Basically, when the phage attacks the bacterium, the bacteria at the population level, they restrain the phage in the beginning until the bacteria reach a very, very high number of density.
Okay.
Because in this way, so instead of having let's say a one-to-one infection, you have one phage, it attacks one bacterium cell and it causes lysis. The bacteria restrain the phage within the cytoplasm of the bacterium. The bacteria replicate a lot in order to increase their density.
Okay.
This is a mechanism basically for the bacteria to increase the probability that the phage might be lysogenic, so be incorporated in the bacterium rather than killing it.
Mm-hmm. So it sounds. So Xavier, tell me if this sounds to you like this is sounds like, to me like it's going to be an attack rate that's essentially something like an attack rate that has a nonlinear response to bacterium density.
Yeah,
which is
that
doable.
sounds right. Yeah, which is doable, but then it's another parameter. So we should know which bacteria does it, so we don't put the potential or that other parameter to all the bacterias.
This phages are relatively specific, so we probably only have to worry about it for a few interactions. So it seems it is another parameter, but it's not one that brings me to tears yet. It's just a I think it's
So
probably we'll read that paper. Yeah.
May I ask you? That's the final question because I'm conscious of time. So, is our hypothesis that the interaction is purely prey-predator? So, is it necessary? So, is there a different type of models if it wasn't? Because we know with phages that we have the lytic phages, but we also have the lysogenic phages, which in a way have symbiotic relationships with the bacteria. They don't kill them. So, is it something that we can understand through our experiments, or do we need from the beginning to take into account that, or decide that this is prey-predator or not?
No, I think we can do it. We need to think about. Yeah, we need to think about exactly what those relationships are going to look like. If it's not a predator-prey interaction, we could incorporate phages directly essentially into the competition matrix.
But if we could reduce the attack rate up to a point that it has a synergetic effect, and it's actually increasing the prey population, and it should be a non-linear one. So we will have a beta of that will be function of the prey as well.
Right. I think it's doable. I think this is actually coming into an area where this is it's an ecological interaction that I'm not familiar with, but I don't see any inherent reason that we couldn't include this in the model. that we couldn't include phages in some sort of way other than as predator and prey. Again, since they're presumably this interaction is also relatively specific, it should be doable. Yeah, it's just an issue of fitting the model correctly, and everything we talked about for fitting the model should work for arbitrary relationships in general, arbitrarily complicated relationships. So I think it's doable.
Yeah. So maybe one of the next webinars, Nina can actually tell us about more details about the interactions between phages and bacteria. It will be very interesting to to understand better what is known about specific interactions. Guys, do you want to have some kind of closing, or so Tucker and Javier before I wrap up?
Thank you for your attention. I know it's not your field and you've been really patient. Thank you.
Thanks, Trevor.
Okay. So I would also like to thank you very much. I think it was really great to have you here and see you and listen to you and interact this way. Very, very exciting from my side, at least, all this information and about the potential of exploring it. I liked also the setup. Thank you, Dora, for preparing this webinar. I think it went great forward. No problems. It was easy to set up. We had basically all the teams participating, so I am delighted. We will try to organize the next one next month, or in a couple of months. We will have a pace to discuss with the groups about what is necessary. And well, have a nice rest of the day.